Thank you very much for the opportunity to present my work. I actually come from Hillel's lab in Tel Aviv University, so we know a lot about my work. And so we are facing an ancient question. So we are all talking about hydrotropism, about the ability to sense, the root to sense water and to grow towards water. But in fact, we actually for a long time don't know what is the signal that governs hydrotropic response. So Darwin was the first to understand that the mechanism of sensing changes in moisture gradient in the vicinity of the root tip resides in the root tip, and postulated that there is a signal that it is originated from this tip and transmitted upward, shootward, to the bending zone. In the case of Arabidopsis, we call it the elongation zone, the place where the bending occurs. Now, since the default growth of a root is towards Earth gravity center, we cannot separate between hydrotropism and gravitropism if we would like to understand the signal that governs hydrotropism. And so if I would like to summarize the relations or the interaction between the two opposing tropisms of gravitropism and hydrotropism, logically, people would ask, what about auxin? How auxin affects hydrotropic response? And the answer is that auxin is not required for hydrotropism as it is required for gravitropism. Auxin is being asymmetrically redistributed across the root in order to promote gravitropic response. But in hydrotropism, it was not detected. So auxin is considered as not important for, or as not as the signal for hydrotropic response. Another potential candidate for governing hydrotropism is reactive oxygen species, which are known to be a secondary messenger. In gravitropic response, they act downstream to the auxin signal. But in case recent findings that we published in our lab show that, in fact, they are important for tuning between the two opposing tropisms. It is important to promote gravitropism, and when hydrotropism is needed, when the direction of growth should be towards water, it actually signals the root, now you are not performing gravitropism, you are growing towards water or bending towards water. So ROS are important for tuning, but as the signal, we cannot consider them as the signal for hydrotropic response. Another logic candidate is abscisic acid, which is known as the major stress response or stress-related hormone, and a very nice work that was published recently by Daniela that is sitting here, and will be, she elaborates more on this on Thursday, so everybody are invited. I didn't do anything. I promise you. No, we need to. I didn't press anything. So ABA was not actually demonstrated to be asymmetrically distributed across the root in response to hydrostimulation. There was no signal from the tip to the elongation zone of ABA. So ABA is important for, or is involved in hydrotropism, but it cannot be considered as the signal that governs hydrotropism. So what could be the signal? Well, accumulating evidence in the literature propose calcium as important for both tropism, for both hydrotropism and gravitropism. So we actually focus on our research in trying to understand the role of calcium in hydrotropism. So in order to study hydrotropism, you need to hydrostimulate the roots. We are using Arabidopsis roots to further understand the role of calcium. And in order to hydrostimulate Arabidopsis roots, we use two well-established and commonly used systems. The first one called split agar sorbitol system, in which basically we grow the roots vertically, cut the medium in a triangle shape just as depicted here, and attach a complementing triangle containing sorbitol to allow diffusion of the sorbitol to the control medium and allowing the diffusion of sorbitol to form a moisture gradient. And another system is the dry chamber system, in which we basically grow the plants the same way we did here. We cut a slice off the agar containing the seedlings, place it in a petri dish on the plate itself, on the wall, such that the root tip bolts 0.5 millimeter into air, pour a saturated calcium chloride solution at the bottom, close and seal the plate, allowing a moisture potential gradient to be formed from the solution towards the gel, forcing the roots that grows into air to bend into gel, because the gel contains water and the gradient is formed towards the gel. So in order to understand whether calcium is involved in hydrotropism, we first check the effect of calcium depletion on hydrotropic response. Under controlled conditions, when you grow the Arabidopsis plants vertically and you attach the sorbitol in this side, you can see that the root tips bend away from the sorbitol, performing hydrotropic bending. Now, the amazing thing was that when we pre-treated plants with a calcium chelator, a cell-permeant calcium chelator named BAPTA-AM, the hydrotropic response was arrested. So we first got an evidence. I'm not pressing anything. We got first evidence that calcium is important or involved in hydrotropism. And in order to study the effect of elevation of calcium in the cytosol, we used a calcium ionophore. And we actually got, in contrast to the arrested response of the depletion, when we artificially elevated the calcium level in the cytosol, we observed an enhanced hydrotropic response, suggesting that elevation of calcium in the cytosol enhances hydrotropism and is important for the hydrotropic response. Now, in order to study the accumulation or the pattern of calcium accumulation in the roots in planta, we use the chameleon sensor. Basically, it's a fret-based sensor in which, upon binding of calcium, there is proton transfer, which allow us to detect and to quantify and even get molar concentrations of calcium in the tissue that we examine. So the signal based on the chameleon presented here under controlled conditions, we took the whole system of hydrostimulation to the confocal by minimizing it, by minimizing the split agar system into on-slide, as we call it, system. It's very, very small. This is a slide that we take to the confocal. And under controlled conditions, we can see an accumulation of calcium, high accumulation of calcium at the tip, at the columella and the meristem. And you can see here an internal tissue of the vasculature. Now, what's happening if we hydrostimulate this root? Let's hope it's going to work. So after 10 minutes, 20 minutes, 30 minutes, we can see an asymmetric accumulation of calcium in this inner vasculature tissue at the forming convex side of the root. So we can actually observe an asymmetric distribution of calcium in the hydrostimulated roots, suggesting that calcium could be, or strongly suggested, as the signal for hydrotropic response. Now we also quantify it, and as you can see here, we are actually facing here a question of what comes first, the signal or the bending. So in order to answer that, we quantify the signal, and we, as you can see here, we have the concave side of the hydrostimulated roots and the convex side of the hydrostimulated roots. And here you can see the curvature. And the curvature occurs right after the asymmetric accumulation of calcium at the bending zone occurs, strongly suggesting that this asymmetric calcium signal is important for promoting the hydrotropic bending. No, it just doesn't work. אני לא הולך להשתמש בו. So another important question that was on the agenda is, which tissue are we dealing in here? We saw an accumulation in a mysterious inner vasculature tissue, and transverse sections of the root expressing this chameleon were not good enough, or didn't have enough resolution to understand in which tissue we are dealing. So eventually we came out with this solution of imaging the roots in two angles of the root in response to in regarding to the phloem and xylem poles, and superimposing bright field images onto the ratio images revealed the xylem. You see here the xylem, the coily tissue, and the phloem, which contains the calcium signals. And this is quite logic due to the nature of sieve elements, which are known to be capable of holding and maintaining high levels of calcium. Another question is whether we're dealing here with a general global response to osmotic stress. We are exposing the roots to high level of sorbitol. It could be that it's just a signal that happens as a general response. And in order to further understand that, we subjected the seedlings to a horizontal split agar system. Here's the normal diagonal sorbitol system, allowing the diffusion in a single direction. And when exposing it to a horizontal split agar, we're actually exposing the root from all directions in a symmetric diffusion of sorbitol. So, and the amazing thing was that when we exposed them to this diagonal sorbitol system, we didn't see any asymmetric distribution of calcium. In contrast to the asymmetric signal that we see when we expose it to the diagonal system, clearly suggesting that this asymmetric calcium signal is important for hydrotropism, and we are not dealing here with a global effect. So surprisingly, after efforts of long years by many or few groups to find genes which are associated with hydrotropism, using forward genetic approaches, only 2 genes were found to be associated or ahydrotropic, not responsive to changes in moisture gradient, and named MEIS1, which is in Japanese Mizu Kusei, hydrotropism in Japanese, and MEIS2. MEIS1 is have unknown function, at least until our paper, and it is known to be associated with the ER membrane. It is known to be associated with the membrane. Remember this is important, and it's hydrotropic. It expresses at the lateral root cap, root tip, meristem, and stele and cortex. And MEIS2, although it is known to have a known function, it is not clear how it is related to hydrotropism. So you would ask yourself, how is the calcium accumulation in hydrostimulated hydrotropic mutants such as MEIS1? When we hydrostimulated the MEIS1 mutants, after 40 minutes, 50 minutes, at that time in Colombia, 0 background, we observed the signal, and you can see there is nothing organized in the elongation zone of the MEIS1 mutant, clearly suggesting that MEIS1 is important for the formation of the calcium asymmetric signal important for hydrotropism. To further understand the mechanism that of forming the calcium signal, we pretty much challenge all the mutants of calcium transporters and signaling involved proteins that we could put our hands on. And we observed an interesting phenotype of enhanced hydrotropic response of plants that were mutated in an ER-associated calcium ATPase that transport calcium from the cytosol to the ER. And when we observed the accumulation of calcium in the background of this transporter mutation, we observed that it have higher accumulation of calcium in the tip and in the elongation zone, suggesting that it is almost ready for being hydrostimulated and to respond to differences in moisture gradient, and explain the rapid response to hydrostimulation. To further understand the involvement of ECA, the ER calcium transporter in hydrotropism, we challenged Colombia 0 wild-type plants with the CPA, cyclopyrazonic acid, which is a specific inhibitor of ECA1. And we observed an enhancing effect of hydrotropic response in plants that were treated with this ECA inhibitor, mimicking the ECA mutation. And in order to further understand the interaction between our protein, that when we mutate it, we get an hydrotropic mutation, and ECA1, MEIS1 and ECA1, we can actually see something really interesting. Treatment with CPA of Colombia 0 resulted in an elevation of cytosolic calcium all over, in the tip, in the meristem, and in the elongation zone, of course. And the elongation zone. And in the background of MEIS mutation, treatment of CPA did not cause any different accumulation of calcium. We can still see low accumulation of calcium, as we can see in the control plants, clearly suggesting that MEIS1 is important for this ECA1 role of forming the calcium signal. MEIS1 is important for the evolving calcium signal formed by ECA1. Now, another important role question that we had is how a signal that is located, yes, how can we explain that a signal that is located in an inner tissue causes bending? Because if a bending occurs, the signal should be somehow mobilized to the outer layer and then cause or result in differential growth of opposing cells. So in order to study that, we needed transverse sections of the chameleon expressing roots. We use for that light sheet microscopy, which allow us higher penetration and less toxicity and all kind of features that we could not achieve using confocal microscope. So to bring it to a light sheet microscope, I actually had to grow it in a tube, and then performing the hydrostimulation in a tube by pressing the diagonally cut tube into a block of sorbitol. And what we saw is that under control conditions, equal of calcium was observed across the root. But in hydrostimulated roots, we saw mobilization of calcium towards the outer layer of the convex forming side of the root to promote elongation and then bending. Immunoprecipitation using ECA specific antibodies, anti-ECA specific antibodies, to study whether there is an interaction between MEIS1 and ECA1 revealed that MEIS1 precipitate with ECA1. So in planta, we see interaction between ECA1 and MEIS1. When we use mutated MEIS1, we saw much less precipitation, clearly suggesting that a fully active MEIS1 is required for interaction with ECA1. Also in yeast, in a heterologous system, using split ubiquitin assay, we saw that MEIS1 interacts with ECA1, but ECA1 does not interact with the mutant. So the mechanism of ECA1 and MEIS1 is important for forming this calcium signal. In order to understand the effect of MEIS1 on ECA1 activity, we use a specific yeast line which is sensitive for depletion of calcium in the medium. And when we express ECA1 in this, when we express active ECA1 in those sensitive yeast line, we can see that it survived and it rescued the phenotype of sensitivity to calcium depletion. Expression of MEIS1 and ECA1 did not allow the rescue, clearly suggesting that MEIS1 inhibits ECA1 activity. And when we express it with the mutant, we can see that it less inhibits the ECA1 activity. So MEIS1 and ECA1 are important for this formation of calcium signal. To further understand the ECA1 expression, we created transgenic plants that express the ECA1 under the promoter, the native promoter of ECA1 fused to GFP. We can see that ECA1 is expressed in the tip and in the vasculature, clearly suggesting in the phloem, clearly emphasizing how important it is in those tissues in which the signal, the hydrotropic calcium signal, is formed. So just to conclude, calcium. We propose calcium as the main signal for hydrotropic response. Calcium is accumulated in hydrostimulated roots in the tip and then promotes a propagation of calcium towards the elongation zone where it is asymmetrically distributed across the root and promote bending. The mechanism of forming this calcium signal is performed by ECA1 and MEIS1. MEIS1 probably binds to ECA1, inhibits its activity. Calcium is less transported to the ER, and the calcium level in the cytosol elevated, and then the signal is formed. And that's it. Thank you very much.
So we have time for one or two questions. Yeah.